Our next session focuses on engineered cell therapies. Please welcome Doug Doerfler.
Well, good morning, everyone. Hope you can hear me. I'm Doug Doerfler, founder and CEO of MaxCyte, and I'm calling in from our Annapolis, Maryland office, so not that far away. But we are just so proud to be a long part of this community, kind of a lifetime member of the Maryland Tech Council and the Maryland Life Sciences Group. I want to give my personal thanks to Martin Rosendale and Brad Stewart for inviting us and organizing this terrific conference today, and of course all the staff at the Maryland Tech Council. So thank you very, very much. Cell therapy started in Maryland. A lot of people really don't know that. It's nothing new to us, and we're going to talk about this industry and how it's really naturally progressed over the last several decades. And on just a little bit of history here, in September of 1990, at the NIH, Dr. Michael Blaze, who many of you probably know, ushered in really the new area of cell-based therapies with Ms. De Silva, who was a 4-year-old patient. She received a transfusion of her own white blood cells, her T cells, that had been genetically altered to carry the healthy ADA gene. And several months later, a second Ohio girl, 9-year-old Cindy Cutshaw, also suffering from ADA, underwent the same procedure. And as a few years ago, as far as we can tell, both ladies are appearing to do well and leading a really relatively normal life. So it's a really exciting time for our industry, but I also believe that it's a really amazing time for Maryland to see the progress that we've been making over the last several decades, maybe 3 decades around the world. And we've seen the next generation of cell therapies, obviously the Descartes and Kymriah, and then we've got another generation of products that are actively and broadly underway. We're going to talk about some of those today with our esteemed panel. And we've got a growing number of therapeutic cell types, novel engineering modalities and approaches, improvements in cell engineering. It seems like every couple months there's a new novel cell type or approach that we're taking to really develop these more complex therapeutic constructs. And as we provide more tools, as the industry provides more tools, I think it really allows researchers and developers to really think outside the box and create even more complex therapies. And so we call it cell therapy 3.0, if you will. And we're seeing that this new class of cell therapies are moving into solid tumors in addition to blood cancers, are moving into autoimmune disease, they're moving into genetic disorders. So a lot of really exciting times. A little bit about the funding environment. And again, we just couldn't be more excited. I think all of us should be so excited about what's going on. First of all, these are data that we've collected from a number of different sources, including ARM and some private sources. But there's approximately 700 genetically modified cell therapies in development. We look to double that in the next 3 to 5 years. Again, many of those are going to be talking about today. The funding environment has been amazing. So in the first half of 2021, there was over $14 billion that was raised in the field of cell therapy. And that's about 70% of the $20 billion that was raised in 2020. So that's a 35% increase over the same period in 2020. There was 20 cell and gene therapy IPOs in the first 3 quarters of this year, and private placements totaled over $1 billion, and venture financing was $5.4 billion, which is a 77% increase over the same period last year. Why is this all happening? Well, I think it's because of the amazing work that's being done, certainly by some of the groups on this call, but also by the pioneering work being done in Maryland. And it's expected that the next generation, in most cases, non-viral cell therapy, those products will be commercially available in the 2023-2024 timeframe. And we predict that when that happens and that box gets checked, we're going to see, again, just a huge amount of capital coming into the space that ushers in a new generation of cell therapies. We have exceptional companies in Maryland, and institutions that are actively involved in cell therapy. NIH, NIST, Hopkins, Maryland, companies like Rooster and Catalent, Cartesian, Regenix, Windmill, Lentigen, Precigen, Kite, Autolus, Arcellus, and AGT. And it's fair to say that we are very, very well positioned from the innovation and very early development to translation to now clinical and commercial phases for these development of the next generation of cell therapies. And we're seeing companies coming into the space. We're seeing an influx of very, very talented people coming from local institutions and from outside the geography to come into Maryland. So it's a really, really exciting time. So really thank you for your support of the Maryland Life Sciences and your participation in this year's conference. And now for the cell and gene therapy track. We have 3 exciting sessions this morning. The first is engineered cell therapies, the increasing complex product development. The second is the cell and gene therapy CMC and manufacturing hurdles and solutions. Should always be a really interesting part of today. And then a little bit about what we talked about earlier, cell and gene therapy, what's on the horizon beyond oncology. So moderating the first session, I'd like to introduce my friend and colleague, Dr. Sarah Meeks. Sarah is the Senior Vice President of Business Development at MaxCyte. And Sarah, take it away.
Thank you, Doug, and thank you for that great introduction, and thank you to the organizers today. For our first session, as Doug described, the development of next-generation engineered cell therapies is expanded into novel cell types and are designed with increasingly sophisticated engineering approaches. In this session, we'll hear from 2 companies developing innovative approaches to these novel cell therapeutics for the treatment of a growing list of intractable diseases. For our first speaker, I'd like to introduce Dr. Michael Klichinsky, co-founder and Senior Vice President of Research at Carisma Therapeutics. His talk is entitled, Harnessing the Power of Engineers Macrophages. Mike?
Thank you, Sarah, and thanks everyone for having me here today. Could the organizers pull up my presentation? Okay, thanks a lot. And next slide, please. Okay, thanks again, everyone. I'm Mike Klichinsky. I'm the co-founder of Carisma Therapeutics. We're not based in Maryland, but we're nearby in Philadelphia. And the company is based around the development of chimeric antigen receptor macrophages and monocytes for cell therapy and oncology. As I think most of you know, chimeric antigen receptor T cells have been truly transformational for blood cancers, leading to significant complete response rates in patients with leukemias and lymphomas that previously had no options. But as this therapeutic modality has moved towards solid tumors, regardless of the target antigen or the type of disease or the route of administration or the method of engineering the chimeric antigen receptor or the T cells themselves, responses have been very limited, where we're going from 70–80% complete response rates in blood cancers to 0 or near 0% durable responses in any solid tumor evaluated to date. So there's a clear disconnect between this powerful technology and responses in solid tumors. Next slide, please. While the exact mechanisms of resistance that solid tumors employ against cell therapies are not clear, there are several well-established barriers. The first being trafficking. Solid tumors do their best to limit infiltration of T cells. If the T cells manage to get in, they're faced with several modalities that seek to suppress those T cells within the tumor microenvironment. There are immunosuppressive cytokines and ligands, potential for T cell exhaustion and energy, and metabolic challenges that limit the proliferation of lymphocytes. And if the T cells manage to traffic into the tumor and overcome suppressive mechanisms, they're faced with the underlying issue of target antigen heterogeneity, where if you go after a single target successfully, you will rapidly select for antigen dim or antigen negative cells that will outgrow and lead to antigen negative relapse, which even in the leukemia setting with CD19 CAR T cells, we know is responsible at this point for approximately 30% of relapses. So we need to overcome these challenges to make a dent in solid tumors. Next slide. Now, it's been known for decades that macrophages are the most abundant tumor-infiltrating immune cell pathologists looking at biopsy slides and so on have recognized that macrophages are abundant in tumors. And we seek to take advantage of the fact that tumors are tropic for macrophages and engineer the cell that they like to recruit in a sort of Trojan horse approach, and to trick the tumors into recruiting these macrophages, but have them armed with chimeric antigen receptors, have them polarized to an anti-tumor pro-inflammatory macrophage phenotype, which we'll come back to, so that once they infiltrate, rather than helping the tumor grow, as macrophages naturally tend to do, they can in fact suppress tumor growth and initiate an anti-tumor immune response. Now, given that macrophages are naturally abundant in solid tumors does not guarantee that after intravenous administration, they'll find their way there. And we and others have in fact demonstrated that if you adoptively transfer macrophages into mice bearing solid tumors, you will see a significant level of infiltration of macrophages into the tumors, and other academic groups have found this to be true as well. Next slide. Now, the CAR-M field, as we call it, is significantly younger than the CAR-T field or even the CAR-NK field. And the primary reason for that is that these cells have historically been very difficult to engineer. Commonly used tools to engineer T cells, such as lentiviral vectors, retroviral vectors, are simply ineffective on macrophages. They have a variety of mechanisms of resistance. Other non-viral modalities, such as DNA delivery, tends to be very toxic and inefficient. mRNA delivery can in fact be very efficient, but given the nature of mRNA, the expression is transient, which for some payloads may be useful, but for chimeric antigen receptors in the macrophage, we think persistence is critical. So next slide. Carisma has established an engineered myeloid cell platform where we have two proprietary viral vectors that are unique in their efficiency in engineering human monocytes and macrophages. The first is Ad5F35, which is a chimeric adenoviral replication deficient vector that has tropism toward myeloid cells. And we've shown we can engineer these cells with routinely over 90% efficiency with no impact on viability, no negative impact on viability. And we also are developing a vector called VPX lentivirus, which is a lentivirus that carries a molecular trick developed by Ned Landau out of NYU that overcomes myeloid cell resistance to lentiviral transduction. Carisma has also established a chemically modified mRNA technology optimized for myeloid cells for non-CAR payloads. And we've also optimized the ability to gene edit monocytes and macrophages using CRISPR-Cas9 to delete genes that may enhance the product profile or the efficacy. For the rest of the talk today, I'll be focused on our lead program, which is Ad5F35 engineered macrophages expressing a CAR. And these macrophages are polarized toward an M1 anti-tumor phenotype. But I want to point out that this platform enables significant optionality in that we can fine-tune macrophage phenotype toward an M1 or an M2 phenotype, M2 being anti-inflammatory, which we don't want in oncology, but of course has potential in inflammatory and fibrotic diseases outside of oncology. Next slide. So the mechanism of CAR macrophages is unique compared to other cell therapies. T cells and NK cells kill primarily through lysis. They secrete lytic factors, and they can induce apoptosis. But macrophages are professional eaters. They can consume entire cells. They can consume debris. They can consume protein plaques, and whatever they eat, they degrade in the acidic and in the acidic proteasome. Sorry, phagosome. So the mechanism of CAR macrophages is as follows. First, macrophages accumulate in tumors naturally downstream of myeloid cell recruiting chemokine gradients. Once they infiltrate, these cells are armed with a CAR, and we demonstrated that putting a CAR in a macrophage can tell these cells what to eat. If the target cell expresses the antigen target, it will get phagocytosed by the CAR macrophage in the absence of any additional opsonization. You don't need antibodies, you don't need complement, you don't need blockade of do not eat me signals. You get robust CAR-mediated killing. In that process, the macrophages are producing pro-inflammatory mediators, cytokines and chemokines, that are recruiting T cells, NK cells, dendritic cells, and other immune cells, and reprogramming the surrounding environment from a suppressive tumor to an inflamed tumor. And critically, unlike T cells or NK cells, macrophages are professional antigen-presenting cells, one of the three in the human immune system, along with B cells and dendritic cells. And macrophages are able to prime a T cell response against secondary neoantigens. So while only target-bearing tumor cells are being phagocytosed, the downstream immune response is broad against patient and tumor-specific neoantigens. Next slide. Now, I'll quickly go through a little bit of data. We demonstrated that we can engineer human macrophages with high efficiency, and interestingly, in this process of engineering with this vector at 5F35 to deliver the CAR, we also found that this vector primes the macrophages toward an M1 pro-inflammatory phenotype by triggering various viral recognition receptors that these innate immune cells naturally possess. Next slide. And we found that by putting a CAR into a macrophage, we redirect multiple macrophage effector functions. Firstly, if the target is present, the CAR macrophage will secrete CAR-induced pro-inflammatory cytokines, such as TNF-alpha and many others. If we evaluate their ability to phagocytose, we can see that CAR macrophages, but not control untransduced macrophages, will eat target-bearing cells, such as HER2-positive cells in this system, but not HER2-negative cells. So they are specific to target-expressing tumor cells. And these combined functions of cytokine secretion and phagocytosis lead to a potent killing function. These macrophages are potent killer cells. And if we look at a tumor killing assay on the right-hand side here, where the y-axis represents tumor growth, you can see that tumor alone and untransduced macrophages have, of course, no anti-tumor effect, while CAR macrophages rapidly kill off all of the tumor cells in approximately 24 hours. Next slide. And this is a video, so if you can just hit next slide and they'll play. So what we're showing here, if you could hit next slide, and hit the other video too. There you go. Thank you. So what we're seeing here is a real-time phagocytosis assay where we label the target cells with a dye that only glows while the cells are being internalized by macrophages. And you can see that control macrophages do nothing, while our anti-HER2 CAR macrophages are phagocytosing tumor cells quite actively, and you see this green signal lighting up as the cells are all being eaten within approximately 24 hours. Next slide. Thank you. Now, in addition to effector function, we found that the CAR being triggered by antigen on these macrophages reprograms the macrophage phenotype itself. And using single-cell RNA-seq here, which allows us to dissect the macrophage gene expression profile at a single-cell level, we find that prior to antigen expression and antigen exposure and after antigen exposure, the phenotype of these cells completely changes, and the clustering in the single-cell analysis is completely different, suggesting that the macrophages have changed their nature, they've changed their gene expression. And when we ask what have they changed, we see that the CAR leads to them becoming further activated and further M1 polarized, and they produce a variety of chemokines, which are factors that recruit other immune cells, and a variety of cytokines that are associated with a productive immune response. Next slide. And we evaluated the activity in, at first, immunodeficient xenograft models and found that CAR macrophages, this is a HER2 system, CAR macrophages against HER2 are able to improve overall survival and reduce tumor growth, in this case in a HER2 overexpressing ovarian cancer model. And using humanized solid tumor models, we found that CAR macrophages are reprogramming the tumor microenvironment and leading to an influx of activated immune cells in the TME. Next slide. Now, given that these are macrophages that are innate, these are innate immune cells, their natural role is to interact with T cells and to prime an adaptive immune response. We, of course, have been very interested in characterizing how they interact with a patient's own T cells. Can they recruit T cells? Can they present tumor-derived antigen to T cells? Can they prime a durable anti-tumor T cell response? Because ultimately, a T cell component to the mechanism is necessary to drive long-term and deep responses. And what we found here is that CAR macrophages are able to recruit both resting and activated CD4 and CD8 T cells. They're actively ringing the alarm, calling these cells in. And in an antigen cross-presentation assay to CD8 T cells, we found that CAR macrophages, but not control macrophages, are able to prime and present antigen, or in fact, cross-present antigen to CD8 T cells effectively. So these are not only cells that in theory can present antigen, but these are activated antigen-presenting cells priming a robust T cell response. And we'll look at proof of this in vivo in immunocompetent models in the next several slides. So next slide. Now, toward that goal, to evaluate this in a relevant model system, Immunodeficient mice are, of course, irrelevant as we don't treat immunodeficient patients. We treat patients that have fully intact immune systems. And what we developed here is a syngeneic, fully immunocompetent model to look at the efficacy of CAR macrophages and how they interact with the tumor microenvironment and can they prime the T-cell response. And toward that goal, we established a HER2 overexpressing colon cancer model and demonstrated that CAR macrophage monotherapy leads to significant tumor control and a significant improvement in overall survival, as you can see in the Kaplan-Meier curve on the top right. But notably, looking at the tumor microenvironment, on the bottom left, we were staining for CD8 T cells using immunohistochemistry, and this is a representative image. You can see CAR macrophages lead to a significant influx of CD8 T cells, while the controls do not. So these are relatively cold tumors, and the CAR macrophages are warming them up. They're recruiting not only CD8s, but also CD4s and NK cells and dendritic cells, not shown here. But interestingly, not only do we see more T cells, but we see more activated tumor-infiltrating lymphocytes based on interferon gamma expression within those T cells. Next slide. Now, to put this concept to the test, we developed a model specifically designed to look for epitope spreading, defined as immunity beyond the initial antigen which you target with your therapy. And our CAR macrophages here are targeted against HER2. So what we did was we co-implanted two tumors on opposite sides of the mouse. One expresses HER2. The other is identical in its genetic background. It's the same tumor model, but it lacks HER2. And the idea here is we treated the HER2 positive tumor with our anti-HER2 CAR macrophages, and we looked at the growth of both sides of the mouse. And what we found was that the HER2 positive tumors, or the CAR macrophages, are phagocytosing tumor cells, rapidly melted. But on the opposite side, the CAR macrophages cannot possibly attack those cells. They do not express the target antigen. So any activity we see there must be coming from a broader systemic immune response. And that's exactly what we found. The wild type HER2 negative tumor on the contralateral flank had a significant inhibition of its growth rate, demonstrating what's called an abscopal effect, which suggests there is broad immunity beyond the target antigen. And when we looked at the TME of these HER2 negative tumors, we found the same pattern of an influx of inflammatory cells. Next slide. Now, this is, of course, a contrived situation. Patients don't present with two tumors on opposite sides with complete different profiles of antigen expression. But what does happen is antigen negative relapse. And we sought to model antigen negative relapse by first growing HER2 positive colon cancer tumors in mice, treating them with our CAR macrophages. And we took the complete responders, the mice that were cured, and we held them for 2 months, which is a fairly long time for mice that live about a year. And we then re-challenged them with a HER2 negative version of the same tumor 2 months later, mimicking the idea of antigen negative relapse. And in immunology, these are called re-challenge studies. As a control, we re-challenged naive mice that never had prior therapy. And we found that all of the naive mice died from disease within about a month, while all of the mice that were previously cured with CAR macrophages against their HER2 positive tumors rejected their HER2 negative tumors and were alive and tumor-free out to late follow-up. And when we, not shown here, we demonstrated that this effect was mediated by memory T cells. So together, this data proves to us that CAR macrophages can infiltrate tumors, reprogram the tumor microenvironment, directly kill tumor cells through phagocytosis, they recruit T cells, and they prime long-term T cell immunity against secondary antigens. Next slide. So given that these CAR macrophages are reacting with T cells or interacting with T cells so closely, we of course began looking at the potential for synergy with PD-1 blockade with T cell checkpoint inhibitor therapy. And here we used a solid tumor model that is resistant to PD-1 blockade. So monotherapy has minimal effect. And if you look at this Kaplan-Meier curve, you see in the black curve compared to the control in green, there's a very slight improvement with PD-1 monotherapy. You can see the impact of CAR macrophage monotherapy out to the very late follow-up of 4 months. But when you put PD-1 on top, you roughly double the percentage of animals that completely reject their tumors. And there's also synergistic remodeling of the tumor microenvironment. When you put PD-1 on top of CAR macrophages, you see a greater T cell influx and so on. So next slide. Now with that, Carisma has established a full-scale GMP manufacturing process to bring CAR macrophages to patients. This is the first gene-modified macrophage program to go into man. And we announced earlier this year that the first patient was treated in our open study, and we're very excited to initiate the CAR macrophage clinical field, which I'm sure is to grow. The Process is as follows. Patients first get mobilized to increase their monocyte count. We collect monocytes. We differentiate them into macrophages in a 7-day process and engineer them with our Ad5F35 vector to deliver the CAR and prime them to an M1 phenotype. And the cells are then cryopreserved and shipped to the clinical site in a frozen state, where they are then thawed at the bedside and infused into the patient. Quite analogous to the CAR-T experience from the patient's point of view, except that CAR macrophages do not require chemotherapy conditioning prior to the administration of the cells. The manufacturing time is currently a 1-week process, and we're working towards shortening that even further. Next slide. CT-0508 is our lead program. It's a HER2-targeted autologous monocyte-derived CAR macrophage product that's currently in the clinic. Next slide. And the phase 1 study that's ongoing is an 18-patient study for HER2 2+ with ISH confirmation or HER2 3+ metastatic solid tumors. These are all advanced patients that have progressed on prior HER2-targeted therapies and immunotherapies. And this is a phase 1 safety and manufacturing feasibility study. As I said, this is the first in human engineered macrophage, first in human CAR macrophage. And we're very excited that this study has gotten off the ground. It's currently recruiting at 3 sites, UPenn, UNC, and City of Hope, with other sites coming on board in the near future. And we plan to present the early safety, manufacturing feasibility, and some clinical correlates at SITC later this year. And next slide, and I'll wrap up very quickly here. You can jump forward. One more. So Carisma has a pipeline of CAR macrophage products. CT-0508 is our lead program that's currently in the IB trial that I described. We're working toward opening regional administration studies. We're collaborating with the City of Hope to develop CAR macrophages for HER2 overexpressing brain tumors for regional administration. And as I described, we're evaluating combination therapy with T-cell and macrophage checkpoints, CPD1 and CD47 respectively. And we're also, we've disclosed 2 other solid tumor targets that we're working toward. The first is mesothelin, and the second is PSMA in prostate cancer. And I think I'm at time here, so I'll stop, and thank you for your attention, and thanks for having me here.
Michael, thank you very much. Great talk. To the audience, we'll take questions at the end of the session, time permitting. And now I'd like to introduce Dr. Michael Singer, the Chief Scientific Officer of Cartesian Therapeutics. His talk today is entitled RNA Cell Therapy in and Beyond Oncology. Mike?
Good morning. First of all, I'd like to thank Maryland Life Sciences and the Maryland Department of Commerce. This conference this week is just the latest example of your long-standing support that has created an environment where small biotechs like us can succeed. Could we please bring up the slides? So, I'm a physician scientist by training, and in a couple hours, you'll actually meet my business partner, Murat Kaleoglu, who's the CEO of Cartesian Therapeutics. We met 20 years ago during residency and have worked together ever since then. 5 years ago, we sold our last small biotech company, decided we needed to take a rest, and after 7 days, we started Cartesian Therapeutics. And if we can go to the next slide, please. We started the company with the mission to become a leader in mRNA-modified cell therapy. And today, we're a fully integrated clinical stage biotech company. We're currently in 3 therapeutic areas: autoimmune, oncology, and respiratory. And as I'll show you toward the end, we have the capacity to move into other areas. Now, what does it mean when I say fully integrated? Within the building you see here in Gaithersburg, we have manufacturing and quality, basic sciences and preclinical sciences, clinical operations, and regulatory affairs. And with everybody in the same offices, we're able to react very quickly to new information, iterate on our products and in our approaches to studying them in the clinic. We have currently 3 assets in clinical trials. I'll tell you about that in a moment. But our most valuable asset is the people you see here. We're very proud that we have a high concentration of physicians and scientists, which really helps us develop these products with the clinic in mind. So, and then lastly, we do have, as you see here, a wholly owned GMP manufacturing facility. We not only manufacture our RNA engineered cells in that facility, but we manufacture our own RNA. Next slide, please. So just as we heard before in Mike's talk a moment ago, how a new cell therapy modality can open up cell therapy treatments to new patient populations, this is how we've looked at RNA cell therapy. So to tell you about the fundamental differences between conventional DNA-modified cell therapy and RNA cell therapy, I need to start and just review a few things about DNA therapy. So as you know, this involves genomic modification. It's irreversible. And the cells that are modified with DNA, of course, they can replicate that DNA. They can divide to yield 2 cells that are equivalent to the parent cell. And we normally look at this as a good thing. In fact, we rely on that phenomenon with CAR-T because we typically administer a dose that is subtherapeutic, and we rely on those cells to expand in vivo to reach a therapeutic level of exposure. The problem is that we can't control that expansion. We can't control the speed or the extent of it. And so if you think about this in a pharmacology sense, there are unpredictable pharmacokinetics, and as a result, toxicities are difficult to avoid. So this explains why CAR-T, for example, to date has been reserved for sickest patients, most advanced, most refractory, and limited, for example, to the most advanced cancers. As you know, it's also expensive to modify cells with DNA. It takes a long time. And there's another expense involved because when cells have toxicities, there's all sorts of other clinical expenses involved in caring for those patients. So now let me switch over and talk a little bit about RNA. As you know, RNA degrades naturally, and it has a fixed period in the cell. After the RNA degrades, the cell loses the phenotype that we engineered that cell to have. In many applications, you look at this as an undesired property, but we actually look at this as a desirable property for other applications, and that's because the RNA allows us to effectively limit the copy number of our engineered proteins from the very start. And so our cells are not sensitive to expansion. In a sense then, again, coming back to pharmacology, we have controlled exposure, and as I'll show you, this confers excellent safety to these cells. Because we have excellent safety, in oncology now, we can think about moving toward frontline treatment, where patients are, first of all, constitutionally more healthy, the tumor burden may be lower, the cells themselves may be less resistant to treatments. And in the case of autologous therapies, the cells that we're collecting to modify may be much healthier and more numerous. As you also know, with RNA, manufacturing costs are lower and timelines are better. When a therapy like this is safer, it's much cheaper to take care of the patient in the hospital. In fact, we're administering RNA-engineered CAR-T cells in outpatient settings today. Just to finish on this slide, I'll call your attention to these graphs at the bottom. On the left side for DNA, we're illustrating how exposure to those cells, because of rapid expansion, can move up into a toxic level of exposure. And then on the right, 2 points I want to make. First of all, because we control the dose and copy number from the start, we're able to control the maximum degree of exposure. The other opportunity that we have with RNA is to re-administer the drug so that you really have a treatment paradigm and pharmacokinetics and pharmacodynamics that look more like a biologic drug, for example. So next slide, please. Today we have what we call the RNA Armory platform. This is a series of mRNA and cell biology technologies here in Gaithersburg that allow us to modify effectively any kind of human cell with any number of proteins. As you know, there's no theoretical payload limit on the number of different mRNAs that we can introduce into a cell. And so we take those RNAs, we engineer the cells, we freeze them in vials, and this is key. We can prepare enough cells to allow repeat dosing with a single manufacturing run. Next, as you know, the cell naturally translates those RNAs into multiple therapeutic proteins. Now, let me say a little bit about those proteins. We can introduce RNAs to express a membrane-bound protein or a transmembrane signaling protein. We can also engineer them to secrete signaling molecules or enzymes, any protein that you'd like to administer into the systemic milieu. And we can also express intracellular proteins and even RNAs that are non-coding but have physiologic effects intracellularly. One way to think about these cells is that they serve, first of all, as a factory where we can express any biologics we wish. Second of all, as cells that can also mediate direct effects such as cytotoxicity. And then third of all, as a vehicle that allows us to deliver the therapy to a specific tissue or organ. And we'll talk a little bit about that at the end. So once we have the patient in the clinic can thaw these and simply administer them by intravenous infusion into the patient. And we end up, as I said, with a combination therapy, which we can, if we want, target directly to diseased tissue. So next slide, please. Here's an overview of our pipeline. As I mentioned, we currently have an asset in the clinic for autoimmune disease. This is the first RNA engineered cell therapy to enter the clinic for an autoimmune disease. I'll tell you more about that in a moment. We have a program in respiratory disease. This is the first RNA engineered cell therapy in a respiratory disease. And then, of course, in oncology, we have a variety of autologous programs, including Descartes 11, the first RNA engineered cell therapy in frontline cancer. Over here on the right, you can see that there's, because of the mechanism of action of these products, there's a variety of other diseases that we have an opportunity to pursue in the future. Now, today, you hear a lot about cell therapies in oncology. So today, I think to keep it fresh, I'm going to focus on the autoimmune and respiratory programs, okay? So we can go to the next slide. So as I mentioned, Descartes 8 is the first CAR T cell in autoimmune, and we're developing this for an autoimmune disease called Myasthenia Gravis. Say more about that in a moment. Descartes 8 is an autologous CD8 positive T cell that's engineered to express, with mRNA of course, to express a chimeric antigen receptor directed against BCMA. And we started this program based on technology actually in license from the National Cancer Institute. I'll go ahead and tell you a little more about this on the next slide. So first, we're targeting a disease called Myasthenia Gravis. Now this is an orphan indication in the sense that the generalized systemic form of this disease affects about 60,000 Americans. It's a progressive disease of muscular weakness and paralysis. If patients are untreated, it's actually fatal in about 1/3 of patients. And the problem is, as you know, that a chronic disease like this requires chronic heavy-duty immunosuppression, where over the long run, the treatment can be as bad as the disease. So patients are often on toxic immunosuppressants, not only corticosteroids, but also on azathioprine and mycophenolate, for example, and you're familiar with the side effects from drugs like this. So on the next slide, this is a textbook view of myasthenia. It helps to explain why we chose that as an initial autoimmune indication. So in this disease, autoantibodies directed against particular proteins at the neuromuscular junction that connects motor nerves with muscles are attacked. There's inflammation at that neuromuscular junction, and neurotransmission is impaired, leading to the muscular weakness. So the autoantibodies, of course, come from plasma cells. And the important point here is we're accustomed to thinking about BCMA as as a target for myeloma. But of course, it's a protein that's also consistently expressed on the surface of plasma cells. And we verified this when we went into this program. So the idea is if we can interrupt these pathogenic plasma cell clones from secreting these undesired antibodies, we may be able to treat the disease. So on the next slide, more specifically, what we're aiming to do here is target long-lived plasma cells. This is key because they're generated over very long periods of time, but with a brief punctuated therapy, we can eradicate all of them. And even though the therapy is then taken away, it takes a long time for those cells to regenerate. So this is actually a view of a textbook view of the pathogenesis of myasthenia gravis, and it shows all the various targets where where the field's tried to intervene. So of course, we start with T cells, and I've mentioned the immunosuppressants. There have been efforts with B cells, for example, with rituximab, with mixed results. I'll tell you about long-lived plasma cells in a moment. And then there are efforts to directly attack the autoantibodies, for example, with FcRn targeting therapy, or directly at the neuromuscular junction, either by preventing complement, as is done with Alexion's eculizumab, or simply improving the amount of acetylcholine. The problem, in our view, with interfering at the level of the autoantibodies of the neuromuscular junction is that an enormous amount of physiologic amplification has already occurred. So there's a very high number of antibodies, molecules that we have to block there. So this helps to explain why Descartes 8 doesn't require continual administration or target suppression. And of course, using it is not exclusive to any other therapies. You can treat with Descartes 8 and then use any of these other agents that I've mentioned. So on the next slide, let me tell you a little bit about what we know so far about Descartes 8, because it's in the clinic. We've studied the safety of these cells both in a multiple myeloma population and in myasthenia gravis. And remarkably, to date, we haven't seen any product-related serious adverse events. We haven't seen any cytokine release syndrome or neurotoxicity. And it's important to emphasize that these therapies don't require any preconditioning or lymphodepletive chemotherapy. This is safer and more convenient for the patient. As I mentioned, we can administer it in an outpatient setting. And in a moment, I want to show you the early evidence of clinical efficacy as we're enrolling these patients. So on the next slide, please. This is the first cohort, which we've recently completed in a phase 1 open-label study of patients with moderate to severe generalized myasthenia gravis. There's various ways that disease severity and activity are measured in the clinic. I won't go into the details here, but suffice it to say that there are several validated instruments, including this MG composite score, the QOL-15, and the QMG score. As you can see, they're highly intercorrelated, both in our very small data sample here with 3 patients in cohort 1, but also in larger trials, such as the ADAPT trial. What we're showing in each of these here then is a score of disease activity. Some of the items here are patient-reported, some are assessed by the clinician, and in some cases it's a combination of both. What we're looking for here is a downward deflection reflecting lower disease activity, and in the red tracings you can see a historical placebo control from the ADAPT trial. So these are means that I'm presenting, but the patterns here were seen consistently in all 3 patients. And as you can see, beginning about 2 weeks after administering Descartes 8, we see a reduction in disease activity. A reduction of 2 to 3 points on these scales is considered clinically meaningful. As you can see, that response deepens over a period of several months. And we saw by the end, 3 months, rather dramatic responses. Now, these patients have been followed for differing periods of time, but In this case, we have one patient where we've followed for several months now, had an opportunity to retreat that patient because the response that you see does wear off after a period of about 6 months. When we retreated that patient, we saw exactly the same shape of response. So we look forward to announcing more results when we have a larger number of patients. So on the next slide. Now I'm going to switch gears and tell you a little bit about a program in respiratory disease. This is called Descartes 30. It is completely different. This is an off-the-shelf therapy. It's not based on T cells. It's based on mesenchymal stem cells. And we're developing it for acute respiratory distress syndrome. And in contrast to CAR-Ts, these mesenchymal stem cells in our case are adapted to secrete 2 enzymes. I'll tell you a little bit about why in a moment. So we can go to the next slide. The pathway where we want to intervene here with this product is neutrophil extracellular traps. Now, I had never heard of these in med school because they hadn't been discovered yet. They've only been discovered about 15 years ago. But it turns out that they're a very important arm of the innate immune system. I would venture to say as important as complement. We're just starting to learn about them though. So it turns out that neutrophils, in addition to the other mechanisms we know, undergo a very peculiar kind of programmed cell death where they simply expel their nuclear content on basically in a suicide mission. And the DNA, the chromatin, forms a sticky web that from an evolutionary standpoint has a benefit of trapping bacteria. And the proteins that are on that chromatin also recruit other inflammatory responses. So it's an inflammogenic stimulus. You can see here a picture. In this case, there's a neutrophil shown in yellow, some bacteria in blue, and this neutrophil has undergone mitosis and expelled its green chromatin. So we're learning now that a variety of diseases that have been refractory to all of the other autoimmune treatments that we have appear to have a very important component of neutrophil extracellular traps. I won't go into all of them today, but I am going to tell you about acute respiratory distress syndrome, and a couple more that I'll note where there's major need is in lupus and rheumatoid arthritis. So on the next slide. Acute respiratory distress syndrome. This has come to the forefront because it's the most common cause of death in patients with COVID-19. But of course, there's a variety of other causes, and this is something that we see commonly in a critical care unit even before COVID-19. So on the left, this is a patient, the lung of a patient who died with ARDS, and these black circles show neutrophil extracellular traps deposited in the lung. If you look at on a more microscopic level, we can see that actually the microvasculature is blocked by these nets. And there are some views proving this on the right side, again, in patient with ARDS. So on the next slide, I've mentioned what we've developed to try to intervene here, Descartes 30. Again, there's no need for preconditioning chemotherapy. And let me just take you through this experiment. I think of this as a 4th grade experiment because it has slime, and it's something that any kid can watch over the period of 10 minutes. We have Descartes 30 cells and controls on the left being added to neutrophil extracellular traps, and then all the way over on the right, within 10 minutes, you can see that Descartes 30 cells secrete enzymes, digest the neutrophil extracellular traps, and the control doesn't. So on the next slide, we put this into an animal model. We see that we can prevent the characteristic fibrosis of acute respiratory distress syndrome, and we're now taking this product into the clinic. We'll have more updates on that soon. Next slide. So key takeaways. Because of the unique pharmacokinetic and pharmacodynamic properties of RNA and RNA engineered cells, we have an opportunity to provide a safer cell therapy and treat a whole new set of patients, both earlier and with other diseases that may not be life-threatening but can be very debilitating. And I've told you about Descartes 8 and Descartes 30. So next slide. Thanks very much, and I'll finish again with the picture of our most valuable asset. Thanks.
Thank you, Dr. Klichinsky and Dr. Singer. Thank you, Doug Doerfler. We are unfortunately out of time for questions. I would say if you want to contact the speakers directly with your questions, please do so. And I hope you enjoy the rest of our morning session on cell and gene therapies. And again, thank you for the opportunity to be part of this, this conference over the last couple of days.
Thank you.
This year's Maryland Life Sciences Bioinnovation Conference is sponsored by Kite. Kite is a global biopharmaceutical company that is part of Gilead Sciences. Kite is based in Santa Monica, California, and has commercial manufacturing operations in North America and Europe. Kite's singular focus is cell therapy to treat and potentially cure cancer. As the cell therapy leader, Kite has more approved CAR-T indications to help more patients than any other company. MaxCyte. MaxCyte is an emerging global leader and trusted partner in the biotherapeutic, cell, and gene therapy industries. Headquartered in Gaithersburg, Maryland, we're proud to have supported the local life sciences community for over 2 decades. Our flow electroporation technology and next-generation expert platform helps accelerate, streamline, and improve drug development from research to commercialization. Now listed on NASDAQ with an IPO in July 2021, MaxCyte is attracting new investments to continue its commitment to enable and accelerate therapeutic programs. And Novavax. Novavax is proud to sponsor the 2021 Maryland Life Sciences Bioinnovation Conference. At Novavax, we are relentlessly committed to one goal: developing vaccines against the world's most urgent infectious disease threats. Our team of experts is working in partnership with leading organizations from around the globe to advance our pipeline, including our COVID-19 and seasonal influenza vaccine candidates. For more information about our efforts to combat the pandemic and to see our latest career opportunities, please visit Novavax.com. We'd like to thank these and all of our sponsors for their support of this year's Maryland Life Sciences Bioinnovation Conference.